Okay, so this is the last lecture ever. No, this is the last lecture of the cytoskeleton, and there are not many lectures left after that. So I don't know what happened, but it's going to be a very sad story with cell cycle. Sad, sad. I don't even know what to do. So let's just finish this up, and then if we have extra time, I can answer any questions that have come up. And then just a reminder, please, by end of day Thursday, any exam questions, you need to get them to me by end of day Thursday. Exam is on Monday. Very exciting. I know you guys are real chatty. Everyone's very comfortable with me now. Yes.
There's no recitation.
No recitation next week because Wednesday there's no class. I don't think so, right?
No.
Okay.
No.
Had a moment there. Okay. No recitation next week. Okay, so this is the stuff that you need to know for the exam. So notice that we've been building from major concepts and components and parts, and now we're starting to talk about how things work together. If you're having a hard time integrating all of this, now is the time. Over the next 2 days is the time to get in touch with me. So after going through all those accessory proteins, we started talking about motor proteins and how motor proteins move cargo and how that's regulated. We spoke about muscle contraction the other day and how that's an example of motor proteins working and how that's regulated. And then we talked a little bit about permanent structures versus structures that are built as needed. So permanent structure would be cilia or flagella, and a structure that's built as needed would be a mitotic spindle. And so this is kind of where we ended. So here we're starting to integrate all this information together. So now you understand concepts of, okay, that was you, not me, that was you. Sorry. Sorry, I thought I had a phone beeping at me. So you understand the major concepts of treadmilling and the concepts of critical concentration. And so you need to really understand that and apply those concepts to really understand all of this stuff. And so this is just a nice big picture of cell movement and active polymerization so that the cell can move. And so you'll have some kind of extracellular stimuli, and then you'll have activation of some of these accessory proteins. Here you're seeing WASP and ARP. And then you're seeing ARP will activate by bind, it will become activated, it will bind to the sides of actin filaments, and then it will promote elongation. And then you'll have, let's see, that's D. Then you will have the growing filaments are pushing your membrane forward, and then you're going to have disassembly, capping proteins. Then you'll have disassembly of the older actin. Remember, cofilin likes to bind ADP-bound actin. So your older actin will be in the back here, and it will be bound by cofilin and disassembled. That will create a new pool of monomer that can be added at the leading edge. And so this is this treadmilling that we talked about. That's kind of, rather than the spontaneous treadmilling that happens in a test tube, this is kind of treadmilling that's motivated by, that's promoted by proteins. And so that pushing against the plasma membrane is what's creating these protrusions that are associated with cell movement. Is everyone like, that's one of my favorite pictures. But anyways, yes.
So cofilin actually builds, because it takes things away, right?
Right, so it breaks down old actin so to create a pool of subunits that's available for the new actin polymerization at the leading edge. So that's why it's kind of shaped like an arrow, because it's disassembling back here, and then it's coming up here, and that that growth at the leading edge, at the plus end, is moving the cell forward. Okay, so actin, myosin, and all these regulatory proteins are working together to promote cell colony. That's kind of what I just showed you. There we're just looking right under the cell membrane. But really that's what's going to happen. Here's a cell that's moving forward. It's moving to your right. And here's it's gonna create this protrusion, and that's where your leading edge is. And so all that actin polymerization is occurring over here. But at the same time, this rear needs to move forward, and so you're going to have different type of actin polymerization in the rear, and different kinds of myosin associated there, myosin 2, and you're going to have the contractile actin that's going to contract and push the rear forward. And that's how you get cell movement. Right, so you've got actin polymerization at the plus end in the cell cortex. So here's your leading edge here. So this is what's moving forward, and you're having your actin polymerization there. And actually the rest of your cytoskeletal structures, such as your microtubules and your intermediate filaments, are kind of stuck around the nucleus. The rest of the cell is kind of filled with this actin network that's focused on moving the cell forward. So here we're using ARP to nucleate the actin, and this is occurring at the front of the lamellopodia. So that is at this leading edge right here. So we saw this picture way at the beginning when we started talking about this. We were talking about actin and the function of ARP, I think. And so here I'm just showing it again, that you have red is your actin and green is your ARP. And so it's really, you got that activity of ARP nucleating actin right at the cortex at the leading edge where you're moving the cell forward. So ARP is binding to the sides of actin, and it's the treadmilling that's promoted by ARP nucleating, and then, as you'll see in a minute, the cofilin removing the older actin that promotes the movement. And so this is actually electron microscopy of your actin web. So I keep saying ARP promotes actin web formation. It really is promoting kind of this branched web, because ARP is binding to the side of your actin polymers. So you're getting nucleation at your leading edge, and you're getting depolymerization of your old filaments behind the leading edge by cofilin, and that's what's driving this unidirectional motion. So it's moving in one direction. And so this is showing, so here you have red is your actin, and your green is your cofilin. And so you could see at the cofilin is only present further back. So at the front, at your leading edge, you have your newer actin. If you were standing for ARP, where would you see your ARP? Everyone's going. Would you see your ARP over here? Would you see it over here? Yes. Yeah, right. So you have your cofilin back here because it's busy disassembling your ADP-bound actin, your older actin. So I have this question here, and this is just, we're discussing it now, but when you're studying, I want you to be thinking about this. How is the activity of cofilin driving the treadmilling process that drives the forward movement? So we've kind of just talked about it, but just for the sake of people who don't understand everything as well, so you understand what treadmilling is, right? Right, so who wants to tell me what treadmilling is? Who feels confident? Yes.
The equal assembly, disassembly plus the minus n, and it never changes
Okay, so you've got so you're saying equal disassembly. So the truth is that you'd never change its length. It looks like it's moving. Is it equal assembly and disassembly? No, because that's like when you're kind of steady state. But that's where you're. So let's add on to it. But yes, the length never changes, and it looks like it's moving. Yes. Yep. So you've got this increased growth at your plus end, and you have this disassembly at your minus end, and that's what causes it to seem like it's moving. And that's what's happening here: is you're growing at the plus end, and cofillins is disassembly at the minus end. Yes. Come again. You're so loud in recitations. You're so quiet in lectures. No, I feel like they're different things. Anyways, single you out. Yeah. Critical concentration. The critical concentration is higher at your plus end and lower at your minus end. Actually, I believe it's lower at your plus end and higher at your minus end. But when you're beneath your critical concentration, so you're beneath your critical concentration at your minus end and you're above it at your plus end. So that's, but I don't want, so when you start thinking about this, I don't want you to think that necessarily the disassembly is going straight to the plus end, like going from your minus end to your plus end. I mean, it really what it's doing is creating a pool that's available to go to your plus end. But when you're talking about treadmilling, yes, you're disassembling at your minus end, you're adding at your plus end, and that increasing size at your plus end is what's moving the cell forward because you're pushing up against your plasma membrane. And so how is cofillin driving this? Well, what's it doing? It's promoting the disassembly at your minus end. How are we feeling about this? Good? Simple? Easy peasy? Okay. So this is what's happening. So you've got your net filament disassembly. So this is your cofillins binding here to your older, so this would be your leading edge up here, right? Your cofillins binding your older actin, and it's disassembling, and it's available. And here it's showing it again in an arrow traveling to the plus end, but that just allows for more monomers that are available to assemble at your plus end. Okay, so motor proteins are involved also here. So myosin 2 is going to associate with your actin to pull the trailing edge forward, and also to pull the trailing edge forward. So you've got your leading edge here. So there's your actin, and then you've got your cofillin and your ARB, and you're nucleating, you're pushing your membrane forward. But then at the rear, this rear is trailing. And so here you have a different type of actin forming. And then you have myosin 2 associated and contracting your rear to help bring it forward. Okay. So you might be saying to me, okay, but what's controlling all of this, right? Is that what you guys are thinking? How is this all regulated? No? Yeah, someone said that. So the Rho family of proteins is Rho family of proteins is involved. It's like, where am I coming from? Rho family of proteins are involved in actin organization to generate cell polarity in response to extracellular signals. So really what you need to think about is, okay, so this is some kind of protein that's helping to transmit a signal from outside of the cell to inside the cell that's regulating the different accessory proteins and having an effect on the cell structure. Okay. So One reason, so what do I mean when I say polarity? What does cell polarity mean? Does it mean anything? Lots of water? Yeah.
Maybe that the cells have different sides.
Right, the cells have different sides. So when we talk about generating polarity, when we have a cell that's moving, you'll say you have a front and a back, right? And so what other reason might you need polarity for? Yeah. For function? Give me more. I didn't hear you completely.
One side might have some structure on it, it's necessary on one side, it's not necessary on
So to function as part of a
the
tissue?
line,
Right. Right, so to function as part of a tissue, you're going to have different sides that have different functions. Yeah, yeah.
Does it have anything to do with chemical polarity, or is it just everything?
It's different from chemical polarity. It's not about charge, it's about top and
Just that
bottom.
they're different.
Different sides that have different functions. Yeah. Anything else? Anyone want to add to that? Yeah, I know, because we use so many words that are over and over again, like maps keep coming up and they're all different. Polarity can mean different things, yeah. So also when you think about cell division, you want to have, you need to have some plane upon which the cell divides, so you need to establish different sides for that too. In yeast, when you're budding, there's a need to establish some polarity there. Okay, so I just introduced Rho proteins, and then I'm going to tell you they're part of the RAS superfamily. So what does that mean? What can you tell me about Rho, based on the fact that I told you they're part of the RAS superfamily? Yes?
It's a molecular switch.
It's a molecular switch. What else? Yes?
Responds to external signals.
It's somehow responding to external signals. In this case it is, but not all molecular switches do, but RAS does. Yep. It uses GTP. Yeah. So it's turned on or off based on its GTP binding state. Yep.
Does it have any cap?
It's probably regulated by a gap and a gap. I am so proud of you guys. My children are growing up. That's beautiful. Thank you guys for integrating all that knowledge. Great, so that's all right. So what I'm going to tell you, I guess I need to take a drink. I just want you guys to know, this class has really, it's brought me to drinking Pepsi. I never drink soda, ever, and it's the whole teaching in the afternoon thing. I'm like a dead person. Huh?
So your next step's coffee.
Well, I can't, if I do coffee, I'm done for. I can't do coffee if you're like me. This is like half the caffeine. Anywho. So RAS superfamily of proteins. Rho is a member, yeah. GTP binding, molecular switches, involved in transmitting some kind of signal from one thing to another, activates things, yeah. Okay, so RAC, Rho, CDC42, they're all members of this Rho protein family, and they're all involved in signaling pathways that affect actin organization. So we did signaling, and we learned during signaling that there are these receptors, and they are all about transmitting a message from outside of the cell, inside the cell to affect gene expression, but also there are these receptors that transmit a message from outside the cell to inside the cell and affect cell structure by affecting actin organization. So it might induce cell movement, for example. So these 3 different GTP binding proteins all affect actin differently, and so depending on which one is activated, you'll get different actin structures forming. So this is just showing a cell where they're staining actin, and so this is a normal cell. And here they activated Rho, and that results in stress fiber formation. That's a certain kind of actin polymerization, likely involving hormones, I'm guessing. Then if you activate RAC, this is involved in lamellipodia formation, which is involved in cell movement. CDC42, involved in cell movement, also in filopodia formation. And so you can see by seeing the actin, they're all very different structures that are forming, suggesting that different accessory proteins are being activated or inactivated, different motor proteins are being activated or inactivated by these Rho proteins. Okay? Okay. So how are the Rho proteins affecting actin organization? What are they doing specifically? So their GTP binding proteins are activated by exchanging the GDP for GTP, and then what do they do? How are they affecting the organization? What kind of proteins might they be targeting? You have all the tools in your brain. Yes?
ARP complex.
Affecting 8. Great, ARP. That's one. Okay, what else? Hormones. So those are two of the most obvious ones that are going to result in two different types of actin structures. Right, so we're going to look at a few examples. Right, so first I want you guys all to go back in your brains to that first cytoskeleton protein, that first cytoskeleton paper that we did in recitation about the E. coli and pedestal formation. Tell me when you're there. Okay. Hopefully you guys have all gotten to it, but you guys remember all the figures, right? Okay. So remember WASP was one of these proteins. So WASP proteins are frequently activated by CDC42 and RAC. And then activated WASP binds to ARP and stimulates ARP activity. So this is I love some of these biotech companies have very nice pathway figures, and this is very nice. So this is just the actin nucleation by ARP-WASP pathway. And so you see there's this is integrins, this is some GPCR, this is some RTK, so receptors, right? And then you've got your GTPases, so you get activation of your Rho family GTPases, and then so it could be Rho or RAC or CDC42, and they're going to affect different things. Sometimes you'll get the stress fiber formation, sometimes you'll get the actin myosin contraction, sometimes you get activation of WASP and ARP. Pretty cool, huh? Yeah.
I thought WASP was needed to activate.
Yes, WASP is needed to activate ARP. So it would make sense that one of these Rho family proteins would activate WASP, which then activates ARP.
It's not just in case, because it, I don't know, if it's like ARP, so ARP doesn't need WASP, or does it need WASP?
I wrote enhance. I must have read it somewhere, but WASP is required for ARP activity. I may have written that at some point because maybe there's some low-level activity in the absence. I'm not 100%. So this should sound familiar as you think back to that E. coli paper, where you had E. coli injecting its own receptor, which was then recruiting all of these proteins, right? So remember this? E. coli tier, that was that E. coli protein that was injected into the cell, the host cell, and so it recruits ARP and WASP through NIC to induce pedestal formation. So back then you were looking at this going, okay, yeah. Hopefully now you can look at it again and say, oh, that makes so much more sense, right? So there was this one figure that you guys all looked at where the green, in all of them, the green is your E. coli, and these were in these NIC knockout cells. And so here they're looking for EPEC, E. coli, and here interaction. Obviously, they're going to be interacting regardless of NIC. But here is WASP. So in the presence of NIC, the E. coli was recruiting WASP and ARP. And then in the absence of NIC, it wasn't. And so if you go back here, let's see if we can find NIC. There's NIC. So here's an RTK, and NIC binds here and binds to WASP, which activates ARP, which activates active polymerization. And so that's what was going on. Cool, huh? You guys love it? End of the semester. You can see everyone's getting tired because everyone's starting to do their recitation questions at home. And then they come in and they're like, okay, let's get out of here. Whereas in the beginning of the semester, people are like, okay, like people are like really into it and passionate, and now everyone's just like dragging, including myself. Okay, right, so CDC42, when it's GTP bound, so when it's active, it stabilizes the active conformation of WASP. So WASP can be in two different conformations, active and inactive, and the CDC42 actually stabilizes the active conformation of WASP, which allows it to bind and activate ARP. And so ARP is normally inactive, and then WASP comes along, activates it, and then you get active polymerization. So activation of RAC activates ARP and a whole bunch of other proteins that favor formation of actin webs/networks. And so it's this big signaling pathway, right? So you'll have some receptor up here in the sky that activates PI3 kinase, and then you get activation of RAC, which there's this back and forth there. And then you have activation of WAS, activates ARP, then you get your actin web. It also activates this other protein, which then inhibits this activator of myosin, because you don't need myosin when you're doing the web. That's more for contractile. So you're inhibiting, you're getting less stress fiber formation, and then you're getting inhibition of a capping protein, which you also don't really want if you're trying to move yourself forward. You don't want to cap your actin, do you? No, because you want that treadmilling. And so it's actually pretty cool when you see there's all this communication between all these different accessory proteins to get the active structure that's required for the cell at that time. So activation of Rho, so that was RAC. Activation of Rho results in nucleation of actin filaments by formants, right? So we had ARP and then some of the backside formants, yep. And activates myosin 2, which promotes formation of contractile actin. So before we had web formation, here we have contractile actin bundles. So here you have Rho being activated, which activates formants, and then you get the actin bundle growth, so you get the stress fibers. And then it also activates this kinase, which inhibits this phosphatase, which inhibits this MLC, remember when we learned about myosin and how it could be folded up and inactive, or it could be the light chain could be phosphorylated and it could be activated. So this is this. So you inhibit the phosphatase, which allows more myosin light chain phosphorylation, which increases your myosin activity. Yes.
So the Rho family, since it generates cell polarity, which is like a process on the cell surface, it's like only on one side of the cell, not the other. That's what cell polarity is.
Polarity just means like this side of the cell's going this way and this side of the cell. So directionality of the
So,
cell.
so the row family like makes like a foot that goes out.
Yeah.
With grabs, so is it, in a sense, like promote top?
Yeah, so the row family kind of makes a foot that goes out. I like that.
I was trying to think of a way. I was
Right,
like,
so it's making, the row family is activating other proteins that form these protrusions that are associated with things like cell movement. So that's an example. Right, yeah. So you'll get increased myosin activity which with stress fibers, then you have more contraction, right? And then also this kinase will inhibit this will activate this kinase, which will inhibit cofilin, right? And you don't want cofilin if you're trying to get stress fiber formation and contraction. You want cofilin when you're trying to get the movement and the web and the treadmill, right? Yeah.
It's raccoon, or raccoon is the one that makes raccoon.
Yeah.
Did that, activate the myosin too?
No, it inhibited.
Doesn't
Yep.
Doesn't that need?
So I know where you're going, and we're getting there. Yeah, we're totally getting there in just a second. Because you want to know, because you know the front of the cell is treadmilling, the rear is contracting, right? And you're like, but wait, we're totally getting there. Yep, I hear you. Because I got a nice figure. It'll be easier with a figure. Okay, so polarized cell growth, right? So this is an example in yeast. So yeast, budding yeast. So when cells are growing under starvation conditions, they need to mate so that they could become diploid and sporulate. But don't worry about that detail because we're really focusing on actin polymerization. And so what happens is you have these mating factors, which are signaling molecules that are secreted by cell by a receptor, and form this kind of protrusion, which is actually it's this polarization of the cell called schmooing. I know, scientists. And so this allows for the yeast cell to be able to mate and become diploid. So what happens is the mating factors bind to GPCRs, and then you get the signaling involving these Rho proteins, CDC42, that results in actin polymerization and schmooing. And so in the absence of your mating factor, you just have these yeast cells that look like circles, and then they form these protrusion in the presence of the mating factor. I put that there because that's a show. So there's your this is what's actually happening inside the cell, right? You have a mating factor that's binding to a what kind of receptor? Oh, it's okay. GPCR. Mating factor is binding to the mating factor receptor, activating a G protein, which then activates your CDC42, your Rho family protein. And so then you get activation of Rho and formins, and then you get your actin cable formation and your schmooing. And then you also get, so this is kind of both things are happening, because you're activating Rho, and then you're also, the CDC42 is activating WASP, which activates ARP, which activates actin polymerization, because you're actually forming a protrusion also. So here you actually can see that one member of the Rho family can activate others. And so we'll see this even more shortly. So here, remember we started out talking about cells chasing bacteria way back in the day, weeks ago. But cells moving, chasing bacteria, right? So a neutrophil is going to move towards the source of a bacterial infection, right? And so that's what this is showing here. Here's your neutrophil, and here's a pipette that's leaking a chemical that's found in bacteria. And so the neutrophil is going to move towards the bacteria, right? Just like in that movie we saw. And so this is dependent on the RAC GTPase and activation of ARP. Right, so this is what's happening. So you have your bacteria that's secreting its bacterial things. And that binds to a receptor, and that'll be a GPCR. And that GPCR will activate RAC, which will result in actin polymerization and through ARP, and those protrusions will form through the web, actin web in the front. Because remember, GPCRs, what's their usual type of signaling, right? So normally you'll get some signaling molecule binds to GPCR, and that results in generation of what type of signaling molecules? Does anyone remember long-lived ones or short-lived ones? Remember, we adenyl cyclin, cyclic AMP, calcium release, these kind of second messenger, small intracellular signaling molecules that act very locally, right? So this is going to kind of activate RAC, and that's going to be in the front of the cell. And so you're going to get this ARP activation and this actin web formation in the front. That's going to promote these protrusions associated with cell movement. But then it's also going to result in activation of okay. And so then you're also going to get activation of Rho, but that's going to be in the rear. And so you're going to actually end up with the stress fiber formation and myosin activation and contraction in the rear of the cell. And so actually you're not activating just one thing. Well, I guess it depends on what you need to do. But for cell movement, you need to activate RAC and Rho in order to get movement in the front and contraction in the rear. Is this where you were going?
Yeah, but how would that stay? Like, how would you stay?
Well, since the activation through a G protein usually ends up with production of second messengers, which are localized, they're only going to activate that local RAC, right there. But you're going to get activation through a different type of G protein. Not G protein. I guess so. I guess it is another G protein that's going to activate Rho in the rear. So it's actually, you know what? I see what you're saying. I think what's happening is the activation of RAC is actually inhibiting, activation of RAC in the front is actually inhibiting the activity of Rho in the front. So Rho is actually active everywhere. Well, not really, but it's everywhere, but it's only active in the rear. So that's what's resulting in movement of the cell forwards with the contraction in the rear. That's helping to bring up the rear. Any questions about that? What did I say? RAC activation stimulates polymerization of actin networks in the front, activates Rho in the rear to stimulate actin myosin contraction. Cell moves forward towards the chemoattractant. Okay, but it gets even more complicated than that, surprisingly. Because there's also communication between actin and microtubules, which results in whole cell polarization. So the centrosome, right, so that's where your microtubules are organized, it's affected by the location of your actin. So if you have, so for example, with a T cell, right, so that's a cell that's involved with the immune response, and specifically there are cytotoxic T cells that are going to get close to the target cell and kill, so they tend to kill virus-infected cells, and so they'll get close and actually interact with the other cell. the target cell. And so, so you'll actually end up having the actin polymerization, and that'll actually reorient your Golgi, and remember, your organelles are moved based on your microtubules, and so you'll reorient your Golgi with your microtubules to the location that's closest to the immunological synapse. And so that actually, I feel like Dr. Schneider was telling me that people were getting away from immunological synapses. So I'm going to have to get back to you guys on how current this is. But the immunological synapse is kind of the location where you've got your T cell and the infected cell, or the cell that's being recognized by the T cell interacting and signaling. And so usually, if the T cell is going to kill the infected cell, then it needs to have the Golgi close to the infected cell, so that then you can produce and release all of this, well, I don't know. I'll get back to you about this. I'm not feeling good about this. Okay. This is the last one, pretty much. Okay. So listen, I think that's it for this exam. We still have plenty of time. If you guys have any questions, but I think that's where we're going to stop for right now. I'll keep recording. Yeah.
So, I know you have the so far slide at the beginning of this, but like, what point should we highlight from this lecture? But like, so far.
Okay, so what should you focus on for this lecture? I want you to really start thinking, well, first row is a big one. So this is really about integrating all of those accessory proteins into actual functioning of the cell. So how these row proteins affect the accessory proteins and the result on the actin network in that cell.
About the exams themselves, so we have 3 lecture exams plus the final, and you said we can drop an exam. Does the final count as an exam we can drop?
Yeah. So you only need to take 3 exams in this class. And please do not feel obligated to take the final. Like, I'm not going to be upset if you don't come. No one's going to get you in trouble if you don't come. The rule in the school is that I need to give the final. So I'm giving the final. You do not need to take the final. That is your choice. That is dependent upon your average. Yeah. Any other questions? Yeah. Do we have a quiz? No, I think I don't have one. I think I had planned for one not to be somewhere. No quiz this weekend. No. That makes no sense.
In the earlier question, we were talking about keratin and the bacteria homologs. You were saying that it was just for fun, but it's in the so part. So how much emphasis should we put on this study?
Keratin, so intermediate filament and bacterial homologs. Put as much emphasis on that when you're studying as I did when I was lecturing. So, like, don't stress yourself out, but if it's anything that has the ability to be mechanistic, you know I'm going to lean towards, that's nothing other than recall, right? And so I tend to not ask recall questions, or I avoid them. Yeah. Any other questions? Yep. So, is the final cumulative or will it be on cell cycle? So, it should just be on cell cycle, but I don't know what's going to happen, honestly. I think there will be a handful of integrated kind of questions that cover a lot of things, kind of like how this, and signaling is very related, and signaling, and the other stuff is it's all integrated. Based on the fact that I think there's only a small number of hey guys, let's hold on the chit chat for a minute. Based on the fact that there's only a small number of lectures left, I wonder if I will have to put a little bit more integrated questions. How many do we have after this exam? Like 3?
No, 4.
4?
Because we'll have 2 the week after Thanksgiving and then 2 the last week.
Okay, so 4 lectures. We'll see. We'll see how much I can get to. Yeah, but it won't be cumulative, like, I won't be asking you to recall questions from the first week of class. Any other questions? Yeah.
Do maps bind to the ends or the sides of microtubules?
The question was, do maps bind to the ends or the sides of microtubules? Yes. MAPS stand for microtubule associated proteins. So it's a category of proteins that bind to microtubules. There are some MAPs that bind to the end. They're like plus tips are MAPs. And there are some XMAP215 binds to the end too, right? But so does tau, right? So they're all MAPs. So the way I want you to think about it is there are 3 categories of MAPs. There are MAPs that are involved in organization of microtubules. There are MAPs involved in stabilizing microtubules, and there are MAPs involved in destabilizing microtubules. I guess you could say there are also MAPs involved in, well, that's fine. And so as long as you kind of understand there are those categories and there are some in each category, it should be good. Yeah.
Actually, right off the map thing, tau is, I don't know if I'm remembering this correctly, but does tau's main purpose like block transport?
Tau's
It's like
main,
that
right, tau's main purpose is to bind to the side of microtubules. It could do 2 things. It could space microtubules,
so it's basically tip, okay.
but also it could block motor proteins.
And does map 2 have the same 2 effects?
Yeah.
It'll block the.
Yeah, so there was that data that I showed you where there were a handful of maps that bind to sides of microtubules and they all blocked, I think it was kinesin but they all blocked motor proteins. Yeah, it was organelle movement, right? Yeah.
For spectrin, is that part of the dynactic complex, or is that also considered a linear protein because?
Right. I'm guessing that's the same protein. Spectrin is a linker protein, and spectrin is part of the dynactic complex. So I wouldn't worry about it being one or the other. It is strange that it's an actin cross-linking protein that's also involved in, I don't know. But yeah, so it's both, it does both things. But just think of the dynactin complex, not of all the subunits. I'm never going to say to you what makes the dynactin complex. So just understand the function.
Okay.
Yeah.
Just to make sure, so, vesicles, I guess they can be transported along with actin filaments and microtubules, it just depends on, I guess, the liver protein that's cargo.
Yeah, vesicles can transport along actin and along microtubules. And it just depends on what motor protein it's binding to, what track it's going to walk along. Yep.
So the triple nodule, is that, it caps at the end, the negative end of actin, right? And so that like essentially stabilizes that, and like after that happens, does growth only occur at the positive end then?
Well, so where do we mostly see triple modulin? What kind of actin? We see it a lot in muscle, in the actin that makes up, that's in muscle cells. And so there, you're going to have triple modulin at one end and Hep C at the other end. And that actin doesn't grow at all, right? It's stable.
Because on the side it was like stabilizes, like there was this how like I just didn't put it together.
Yeah.
Okay.
organelles can also move. I think actin and microtubules both can be used for moving, but I think of organelles more along microtubules, but I don't see. I think that they can also move along actin, but we should double check that. Anyone else? Yes.
Could you just explain one more time the negative regulation
Negative regulation of
Same
Staffman
thing I understand,
by phosphorylation. Staffman binds to alpha beta tubulin, right? Okay, and that encourages catastrophe, right?
Yeah, because it like keeps it confined.
Right.
So it just,
Right, so there's less monomer or subunit
yeah.
available. And so if you phosphorylate Staffman and it can't bind to microtubule subunits, then they should be able to readily add. So just, yeah, so it's really about controlling whether or not the subunits are available.
But I wasn't sure, because I know like, when you put negative in front of something, it just,
Anyone else? Okay, well you can go.